it was essentially just cut open the DNA and allowing all the contents of it to spill out. Because the primary agent of the lysis step is SDS, or sodium benzyl sulfate, and that is responsible. It's a detergent. It's like bleach. It's not bleach. It's like dish soap, something like that. It just goes in there, breaks apart the cell wall, and all the information, all the stuff, proteins, DNA, RNAs, just spills out to the solution. After that, you added a alkaline protease. Now, as its name suggested, as a protease, what this thing would do would break down and degrade large proteins. And because it's alkaline, means it happens at a basic pH. Of course, SDS is basic to begin with, so this works at basic pHs, and that's why we use an alkaline protease. The final step here would be the neutralization step. You added the 350 microliters, and the neutralization step is what combines all the garbage together. The proteins, the genomic DNA, the RNAs, they all have those loose ends, have been degraded, and they're being meshed together in a gigantic pellet. It's like when you add, what's the best way to describe it? I can't think of a great example. But like the idea is that the neutralization step forces all these polar ends of all these chemicals to just come together and form this gigantic white bunch of crap. Because it doesn't like the polarity of the neutralization solution. Okay, now that with the neutralization step added the chemical, you would actually invert this a couple of times, so that you get it distributed throughout the entire solution, because it works really quickly and it's a very effective solution. So after the neutralization step, we would centrifuge the chemical, the entire solution, for 10 minutes, and this forces all the physical contents to be pushed to the very bottom of the tube. At the centrifugation step, you would take the liquid from here, because all of the stuff of interest is now in the liquid, and all the garbage, all the big garbage is already out of it. And you would take all the liquid, and you would put it into your spin column. Like that. Now, on the bottom of the top portion of the spin column is this white plate. This white plate is a sulfur-based plate, and what it's supposed to do is gather up DNA when it pushes through to it. So when you put the liquid in there, you're going to spin it again. You're going to centrifuge, I believe, this for 2 minutes, and you would end up with all your DNA sitting in that white plate. Now, attached to the DNA are actually going to be a whole bunch of other things, like proteins and some sugars and stuff like that. So what we do next is the wash steps. We did 2 wash steps. We did one with 700 microliters of a wash, and one with 250 microliters of a wash. And what these wash steps did was push everything off of the DNA, all the extra stuff, the small proteins, anything that's attached to it is coming off of it. So what leaves you behind after the 2 washes, so what leaves behind is your purified DNA sitting on the bottom of in that white plate. So next step was the elution step. Now, we remove this pour out tube because we don't need it, but we put in a new centrifuge tube to prevent, so we can collect all our DNA in a separate tube, and what you did was essentially centrifuge the tube with 50 microliters of nuclease-free water. Now, water has to be nuclease-free because nucleases degrades the DNA, so that just ensures that when you collect your sample, it's not getting damaged or destroyed by any chemical that's existing in the water itself. So you end up with 50 microliters of your water sitting at the bottom of your tube, and with this, we're able to start the next step, which was the digestion.
What you're saying is the nuclease, what is it?
Oh, it's just pure water. It's very, very clean water. The problem is, like tap water and sometimes distilled water isn't purely purified. There are sometimes these small proteins that are called nucleases. Nucleases, as suggested by the EA's name, degrade stuff. So, like the alkaline protease is a protease. It breaks down protein. Nucleases break down nucleic acids and nucleotides. So we just had to make sure that this water is super clean. So when we diluted our DNA, or we eluted our DNA, it'd be preserved. It would be clean, and it would stay in its full shape and structure. No damage whatsoever. Alright, so the next step was the digestion step. And pretty much what we did here was just add a whole bunch of chemicals together. We collected, let's see, it was 2 microliters buffer, 1 of each enzyme, 6 of the water, we took 10 microliters of DNA. Right? 2, 3, 4, 6, 10, yes. So what you did here was take 10 microliters of the DNA you just produced, and you add it together, preferably in this particular order, with 2 microliters of buffer 3, 6 microliters H2O, 1 microliter of restriction enzyme EcoR1, and 1 microliter of the other restriction enzyme Bagel 2. Now we use buffer 3 because that is the ideal buffer for these 2 enzymes to work. And what we did, why we made a 20 microliter dilution, is because the buffer that we originally have in the tube is actually a 10-time solution. We have to get it down to 1 times, and since we're using 2 microliters of it, 10 times 2, 20 microliter solution to make it a nice big solution. We use 1 microliter of each enzyme because these enzymes are actually pretty effective. These are actually extremely effective enzymes. So you saw from the gels how clean and clear they were because they're very good at what they do. Alright, so put them all these chemicals together, and then we incubated overnight at 37 degrees C. For that is the ideal temperature in which these work. There are actually other enzymes that work in other types of temperatures, but 37 is the one we use for these. Alright, so we did the digestion, and that brings us to our final step where we did the gel electrophoresis so we can visualize our DNA. Write that down. Gel electrophoresis. Chalkboard burn on my hand too. Alright, so first thing we had to do was prepare the gel. The gel we made is a 1% agarose in TAE. TAE is the buffer, which is really good for DNA. Water doesn't work. If you put water in a gel, the DNA goes absolutely nowhere. So the TAE allows the current to be carried, and it also allows the DNA to flow through the gel. So that's why we use TAE instead of something like water. we combined to make this sort of gel, so we made a, 50 ml gel. So that means you put 0.5 grams of agarose into 50 milliliters of TAE. Cooked in a microwave for about 2 minutes. So we just heat it till it's boiling. Laying on chalk. So once the solution was produced, we added